Hello, how are you?
Hey, how's your weekend so far?
It's pretty good. Thank you. Happy July 4th.
Yeah, for sure.
Mm-hmm.
Uh, do you have to work tomorrow? Like, do you have a job outside of this right now? Is this like what you do?
um,
Just
uh,
one.
yeah. I sometimes trade, like, cryptocurrency and stuff.
That's cool. I don't know.
Yeah. I do lots of, like, random stuff.
I feel that.
Yeah, I was looking at one of the questions, about a CCD, and I remember doing some research on that when I did, like physics, cosmology research.
For sure. Yeah, like the only bad thing, like, because I majored in like health science, exercise science, like my research is all like practical clinical stuff. So like when I read some of these passages, like I've really had to like
Oh,
learn
yeah.
things. Yeah, it wasn't very helpful. Like it'll help me a lot, like with the patient interaction stuff, but
Mm-hmm.
it's not super helpful for the MCAT.
Oh, yeah. I
I mean,
mean,
I
like,
took all the classes. Like I took genetics and stuff, which helped a lot, but like still, like I don't know.
oh, yeah. Yeah, I mean, like, the research I did that involved a CCD, like, it just so happens that this question involves that, but it was basically, like, using, like, Kepler, which is, like, this probe that we send out to, like, look for exoplanets. And
Gotcha.
basically, it's a way, the project was, whatever. I'll tell you about it some other time,
Okay,
but,
that's good. I was wondering if we could start. I actually was going through the Q, the not Q pack, the bio. No, actually, it's the whatever the CP bank, and I got stuck on one. If we could go through that,
oh, sure.
and then maybe if we could go through a cars passage because
Mm-hmm.
I'm not getting wrecked by cars, but like I finished with like 72nd percentile or something. It's
Mm-hmm.
like I think I'm getting like anywhere from 3 to 5 questions, like 3 out of 5 questions correct per passage.
Yeah, yeah.
I notice it tends to go down during actual tests. Like I don't know if it's just being tired from the first section or the timing, but I have noticed like I have a really like bad habit with like staying on a question too long because I know I can get it right. Like
Yeah, yeah,
if I
yeah.
do go on timed, I can get 90 to
Yeah, yeah,
100% if I
yeah.
do it on time.
Mm-hmm.
Like, but if it's timed, I think I panic and
Yeah.
I do read slow and I answer even slower. So
Mm-hmm.
like the last couple of tests like I took, like I was doing pretty good, but like I ended up like 30 minutes left with like 4 passages to do. So
Yeah.
like I ran out of time and then the stress gets to me and I do even
Yeah.
worse with everything wrong. So
Yeah,
yeah.
yeah. Trust me, I've been there. I've, you know, um, voided my exam, uh, as
I
well.
avoided my first exam due to really bad anxiety and working on it. And then the second one wasn't much better, but I got through it. It's
Oh, yeah,
just,
yeah.
it's
I, I,
tough. So
I
yeah.
voided because I spent way too much time on one problem because am a perfectionist, and then I ended up not even completing like the first section, and I'm just like, F this.
No, it sucks. And like
And then I
my whole
spent like
experience with
the
like school,
time
like,
after the test, like sitting on the bench, like looking like looking it up, trying to figure it out.
oh gosh.
Yeah, it
No,
was an
like I
interesting
got stuck on
question.
the first problem I had. It was like a math thing. I'm like, what
Oh,
the
yeah,
fuck
yeah,
is
yeah,
this crap? I was really
yeah.
upset.
I,
And
I
the
remember a
cars just
question. It
killed
was
me.
about radioactive decay, which I know about, and it was about radon, right? And in the periodic table, there's Ra and there's Rn, I think,
And yeah,
and I
yeah.
I, I chose the wrong one. Yeah, 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 yeah. It happens. It happens.
No, and the problem is like I'm kind of a perfectionist and I can get things right if I stay on it a really long time, but
Yeah,
like they're
yeah,
all worth the same amount. And
yeah.
it's just a problem. I've been working on that a lot.
I
Just like
think
my stress.
I
Yeah.
think like no, like I'm that way too, and I think it's really really like the best way to handle things. But when it comes to like when it comes close to test time, that's when you go into like endurance mode, and that's when you focus on that. I like to separate them, which I think is the best way of doing it because like yeah, if you do it like untimed, at least you can gain that confidence that you are able to actually solve it.
That's true. Yeah, like I really struggled. I was always that student who took the longest on a test. Like I remember my last class I took, like post-batch, was like physics 2, and I had not taken trig. He let me take the class anyway. So like the highest math I took was college algebra. Did not take pre-cal and none of that.
Yeah.
And I got the highest grade on our midterm out of everyone.
Yeah.
But it took me 20 minutes just to beat the anxiety to
Mm-hmm.
get like, because I was like blanking so bad
Oh, I
with the
see.
math.
Yeah,
Yeah.
yeah, yeah.
And then once I got going, like I was good. And
Yeah.
I noticed that happens with my MCAT too. Like once I'm done with CARS, I
Yeah.
feel a lot more confident and I can get
Yeah.
through the biochem and the
Yeah.
psych stuff.
Yeah, that's why I always say it's great to like flag, move on. And like I also like to do like discretes first, cuz like if you find something harder, if you're blanking, right? It always happens usually in the beginning, right? So
Yeah.
so if you like skip and move on and answer some more questions to get some of that momentum, some of that confidence, then you can go back to the thing that you blanked on, because everyone really like the MCAT's designed to psych you out, because pre-meds are like smart, like they've taken all these like really hard classes. They probably have high grades in those classes if they're applying to medical school. So how do you design a test that can, you know, I guess give grades to people who already have high grades, right?
Yeah,
So
that's true.
the first passage, first two like passages, they're going to throw like hard stuff at you. I've seen this every
Okay.
single time I've taken it. Like the first like it's meant for you to just it's meant to just break you down at the beginning.
For sure.
It took me a while to get past that, because I wanted like I you know like I retook like a 518 because I'm like, you know, like I wanted like that, but that when I retook the 518, that meant that it's so easy to get lower than that. So if there was one question that like I couldn't answer, like when I was taking like the practice tests, I would just like end the exam.
Oh gosh.
Cause like, you know, why, like I'm not gonna like take it again and then get lower than a 518, right? Like at that point, like it's like a matter of like one or two, like three or four questions.
Login system work. Yeah, for sure. I just want to get to like a 510, honestly.
Mm-hmm.
Um, yeah, because I did horrible. Like my MCAT was like a 491 or two, something like that. It was really terrible.
Yeah, yeah.
It was CARS. CARS was like horrible. It was like I missed every question. Like
Yeah,
I don't
yeah.
remember reading any of it. It
Yeah,
was
yeah.
terrible. I did really like good on psych soc, but that was like it.
Mm-hmm.
I don't really know why. Just psych soc for me is really easy.
Mm-hmm.
I don't know, but I find it really interesting too. I'm not
Yeah.
real wild about. I don't dislike physics. I'm just not into it. I'm not
Yeah,
really
yeah,
into
yeah.
that much. But yeah.
Yeah,
So I
that
have this. Yeah. So
usually like interest yeah, if you can somehow trick yourself to be interested in things, that's like a boost.
yeah, it just sucks that my friend's in PA school right now. She's
Mm-hmm.
like, "You're never gonna use this physics again." I'm like, I know.
Oh yeah,
All this
yeah.
crap, this will be all this test is like not gonna be there.
Oh
I mean,
yeah.
yeah, some of the physiology for sure. Like, but like, yeah. Anyway, I ranted, but so I was started this passage and I was doing okay and I got kind of mad. Do you see
Mm-hmm.
my screen? Okay. So I kind of know what these are, but I was trying to use the passage to like pick, and I just kind of want to go through a couple of these and like see if like I'm on the right track. So it said the DNMT3A was purified, and it was like just like what is the column chromatography type?
Yep.
So it said the DNMT3A is a methyltransferase modifying the 5 position of cytosine bases. Binds in
Do you
a
know these different types of chromatography?
Yeah, so not perfectly because I know there's like more that are just listed there. I'm not sure if this is right, but like I know affinity binding columns, don't they? I know there's different types of them though, but like
Mm-hmm.
size exclusion, I think doesn't that just separate the proteins by mass only?
So it doesn't have to be proteins. It could be anything.
Yeah.
But well, yeah, it does that. So like just but like think about like how. So like I visualize like a column, and it has like some type of like medium in it, like a stationary like kind of thing. Like let's say beads.
Yep.
Okay, now let's say the beads, like let's say you're trying to purify Na plus from Cl minus. So let's like so let's say that you have like beads that are like positively charged, and you pour in a solution of like sodium positive ion and chloride like Cl minus ion. What'll happen as you pour that? The
Doesn't
beads
it?
the beads are positively charged, yeah.
So I'm trying to like not get this wrong, so I always read this and forget.
Don't
But
worry about
like,
getting it
yeah.
wrong.
So
Just
like,
think of it as exercise for your brain.
yeah. So our station is positively charged, is what you said, and we poured chlorine and sodium into it, right?
Sodium like ion and chloride ion.
Yeah.
Mm-hmm.
Okay. Don't the doesn't the negative charge attract to the positive charge and
Mm-hmm.
bind to the column?
Yes.
So so if it's negatively charged, or wait, did we say it was
Mm-hmm.
negatively charged? The column?
Positive, but you get
Okay,
the
yeah.
idea, yeah.
So that means the chlorine would bind to the positively charged column, and
Yep.
then the
Mm-hmm.
sodium would elute first.
Exactly. Yeah, and you're even using like the right words, like elute and stationary. I just said I forgot what I said, like medium or something. Okay, so anion exchange, cation exchange, those are kind of like it kind of means the reverse, at least how I thought of it. Uh, so basically, like the thing that is changing is the thing that it binds to. So if it's positively charged, it's going to exchange the anion, I believe. But and then vice versa would be the cation exchange. Affinity is just a general term for anything that would have
Yeah,
an affinity. And size exclusion, it would just be like it'll have like holes, I guess, or like
like the pores.
yeah. All right,
Yeah,
yeah,
I know what
you
you're
got
talking
it,
about. So
you
like
got
that
it.
makes it to where like larger versus smaller proteins, well, they can like kind of
Yeah,
determine which one's going to elute or
I
bind
think
to the
like they'll
column.
make the pores like smaller, so the smaller ones lute first. Anything like that. Okay, good.
Okay. So
So
like when I first read it though, like I was thinking because it said DNMT38, it says the DNA methyltransferase. The first thing that came to mind was DNA is negatively charged, but that might not get us to the answer. But I was thinking about that.
uh huh.
Um,
Wait.
and then
Oh, I'm just looking first for um the where they talk
the
about
chromatography,
pure yeah.
yeah. So down here, I did see histidine tagged,
Oh,
and this
nickel
might not,
nitra blah 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 chromatography.
yeah.
Yep.
But I also was thinking, like, not to overthink this question, but since we're talking about it, it said
Mhm.
histidine 6 tag DNMT3A.
Mhm.
I don't know if that's the same thing as affinity, if affinity tagging is the same thing, but I know that like in certain experiments, they like tag something that like fluoresces so they can like differentiate it.
Mhm.
I'm not sure like if that has anything to do with affinity or not, but I was thinking maybe. Um, and then I saw the reducing SDA page after that.
Mhm.
Um,
Yeah. All right. You have the right idea. I think in this case they just used histidine to tag it. Um, but yeah, in some cases it could be tagged with like something that fluoresces. In some
Gotcha.
cases it could be tagged with something that's like radio active.
Oh yeah, radio labeled, yeah.
Yep. Yeah.
But I
You know
didn't
your
see
stuff.
anything in here about anion cation exchange other than like I do know DNA is negatively charged, but like I didn't see anything about that other than they were transferring that methyl group. So I
Mhm.
was thinking maybe it has more to do with the size, but or binding somehow. I don't know.
Mhm.
And I'm trying to remember. I know reducing as there's reducing and non-reducing, and I think non-reducing is the one where it like keeps the like the structural function of the protein the same.
Mm-hmm.
I'm forgetting what reducing is, but I know it has to do with like I know in experiments it has to do with like the band thickness and
Mm-hmm.
all that. I'm
Yeah.
trying not to overthink this. Like I feel like I know a decent amount, but I'm having a hard time
No, no,
like
you're impressing me. You're using a lot of like words that I was like forgetting at the time. So good job. And that's like how you should be thinking about this. Like when you see that, you should be like all these things should be like coming up in your mind. Yeah. So yeah, so for instance, if it's some type of like protein, right, that has like two subunits, um,
Mhm.
and you reduce it, you like break it apart. So instead of like if a protein had like two subunits or whatever, it would be like a large like single band.
Oh
And
yeah,
then
that's
let's
right.
say that you reduce it, you like make it so the subunits come apart, and then you should see two smaller bands that are like lighter, but like combined would be equal to the size of like that big one.
Okay, like say that. Hold on. So say that again. I just want to make sure I get this because I
Mm-hmm.
always forget this. So I know the non-reduce. I can't remember all the rules. Like I try not to remember this stuff, but I feel like it always kicks my ass in experiments.
Mm-hmm.
So like the non-reducing, it retains like the native structure of the protein or whatever. I know the reducing can like. I think there's some denaturation or something that goes on where it like disrupts the structure. But
Mm-hmm.
there's a reason you want to do one versus the other. I'm forgetting why, but I know you're saying the reducing is which one is the one where you'll see two bands visually? Is that the non-reducing or reducing?
Oh well, it depends, but like if you're dealing with something that should normally have like all right, for instance, like in the case of like something with two subunits or whatever.
Yeah.
So well, okay, why don't we use this as an example? This is a Homo Tetramer, right? It means that this has four identical subunits because tetra means four.
Yep.
Homo means well, in this case, it means same. So it's going to have four like subunits, like the protein.
Okay.
So okay, so yeah, so basically, let's say you did some type of whatever gel thing, um, like it should be like let's say like one like big heavy like band, and let's say that you do something to it that breaks apart the like whatever like the tertiary bonds or something
Yeah.
to make those four subunits to come apart, then you should see four smaller lighter bands.
Okay, so that's why it's reducing. And it says to after like showed a single band at 35 kilodaltons. So okay.
Okay,
That makes more sense now. And that also makes me think back to because it said the agarose gel. I
mm-hmm.
think they use that has to do with the charge, but I think they also do that in experiments like PCR and stuff to separate by size though. So it might be size. Part of me is leaning towards A or D, just
Oh
because
wait,
I
you're
don't
saying
know
you're
it.
saying like the gel itself?
Yeah, like the gel agar. The reason they
Okay,
use the gel
so
agarose.
chromatography is going to be different from a gel.
Oh, I'm thinking of. Yeah, I'm trying to like. Okay, why are
Mm-hmm.
they bringing that up then? Like, okay, they use the agarose type of chromatography, not like an agarose gel and like Western blots. All right, I gotcha.
Yeah, I thought agarose was like the thing that um, bacteria can like eat. Agar?
Yeah, it's like pulling my brain to microbiology now. Like,
Uh huh.
yeah. No, I thought they used like in Western blots or maybe it's just PCR. They use that gel. It's like it has to do with charge. I thought it was agarose gel, but it might be something else. Maybe it's. I know the SDS is like the sodium diet something. I'm
Uh,
probably like way overthinking this question, but like it's
no,
making
no,
my
no, no.
brain
This is good. This is
a lot of
good.
different places.
It's good what you're doing because we're not really like focused particularly on the question. We're focused on like the overall like test and like just like how you're thinking.
Yeah.
So good. Yeah. Keep going.
All right. So like if I read the question and went back, I would have stopped like after reading this. I wouldn't have probably gone any farther. Like this just talks about the different conditions, and I mean I don't really see anything in there that would help answer the question. Like that was pretty much it. And then just kind of knowing, which I went through a few questions before this, so I already kind of knew this info. I used it to answer another question. They did talk about binding in a cooperative manner.
Oh, yeah, to DNA. So, like, do you want to talk about that? Like, that's
We
like
don't have. I know
cooperative is just
cooperation.
the
I
same
know
as
cooperation.
like allosteric, kind
Oh,
of.
I know what all that is. I
Yeah.
feel pretty good with that. So I'm just trying to figure out what this is. Is like my brain's like going all over. So
Oh, it's an enzyme.
DNMT3A active as a Homo Tetramer.
Yeah, it's an enzyme, right?
Yeah, so it's a protein.
Yep.
So I'm just like torn between, is it just by size or does it have to do with
Oh,
chart?
okay. So look where it says chromatography, right?
Oh, down here, you mean?
Yeah. So that's what it's going to be. So nickel, I guess, turns into like Ni2 plus, nitriloacetic acid, blah blah blah. So let's see. So so yeah, I always like mix up the anion exchange and cation exchange thing. All right, okay. Now I think I got it. So the thing that the stationary like phase whatever. So okay, let me think. So if it's made of so if it has okay, so if it has a positive charge and it connects to something that has a negative charge, um I think it would be cation exchange because it exchanges it, or no, or it could be anion exchange. That's why like I always like mix these up because it's like it's not as simple as that. I think it might be anion exchange. All right, I'm gonna cheat.
The only thing I was wondering is like DNA is negatively charged. Doesn't that have to do with the binding? And down here they talked about the nickel, so positive, so negative, positive. So would it be maybe
Okay,
cation
sorry.
exchange to negative two?
Oh wait, oh I didn't even see that they were talking about wait with oh okay. So yeah, so all right, the anion exchange is so if the stationary phase is positive, my brain thinks cation, but it's like what is exchanged. It
I can't remember
connects.
that either. I
Yeah,
don't
yeah.
remember
It
that. Yeah.
connects. It's the word exchange that changes it. So if the stationary medium or whatever is positively charged, it's going to exchange that for something that's negatively charged. So that is going to be anion exchange.
Okay, so let me like before I answer like think through this with my brain. So we're going. So the protein is a DNA methyltransferase.
All I looked at was nickel nitriloacetic acid agarose chromatography.
You don't think I need to pull in the DNA? So I just know that fact about DNA like has nothing to do with it. Maybe I guess, but
It's good to know that, but we're just trying to purify whatever that enzyme.
yeah, and I didn't even think the nickel was that important in this situation, just because I'm not really.
Yeah.
Okay, so nickel positively charged. So are we? So are we cation exchanging then? Because nickel is positive or it's the opposite? Like I'm not.
It's that weird thing. It's the opposite. Yeah.
So it'd be anion.
I think so. I hope
A
so.
probably. We'll say it's one of those two. That was A.
Damn it.
Okay.
All right. I hate myself. Okay, let's see. Histidine tagging nickel affinity. Wow, that sucks.
I just think those questions, like some of their questions to me
Hold
are
on.
really shitty. So
No, the explanations are the worst.
yeah.
Hold on. Let me see. Cause. Okay.
I did notice the histidine tagging, like, but I wasn't sure how important that was.
Yeah.
That's why I was kind of leaning towards A for a little bit, but then they kept talking about.
Yeah, I was trying to think of like, all right, let me check out affinity, because I thought anion exchange and cation exchange were versions of affinity chromatography. So, okay.
That seems like a really general, like the most general answer of all of
It
them.
seems like it, but it actually happens to be it happens to be based on a highly specific, like, molecular binding interaction between the biomolecule and another substance. So I mistook it for being like a general term that includes something like anion exchange or cation exchange, but
Okay.
affinity chromatography is just for a specific, like, biochemical that binds to, like, whatever it's supposed to bind to. So
Okay.
so
Circling back to this question though, like,
yeah, so you were right in noticing that it was histidine tagged. So that means that the whatever that, like, stationary material, stationary phase or whatever, is going
mhm.
to bind to that histidine
So
through a very
it,
specific, like, you know, like, very specific binding.
so did they design it to be histidine tag then?
When you
I'm
say
not trying to
design,
overlay think this. This said DNA histidine tag.
yes, yes,
Okay, I was trying to figure out what they were looking for exactly. So I guess they were trying to differentiate
yeah. No,
or separate.
you did the
Okay,
right thing. I went too fast and I just looked at the nickel
that
part.
was good.
Yeah. So
Yeah.
okay. So affinity chromatography, right? It's not just any generalized affinity like I thought. It is a specific, like, biochemical thing. Like, if it was like DNA that's, like, negatively charged, and you had a column full of, like, whatever, histones or something, that would be another example. But it's a very specific, like, binding interaction between a biomolecule and another substance. The
Okay.
biomolecule in this case is histidine, and the other substance is this weird thing.
Yeah, and that was a like to me was kind of a difficult question to narrow down because they were going all over the place with some of these different things. Like,
Mm-hmm.
yeah, okay.
Wait, with what?
Just the fact that they were kind of like, I mean, all over the place with what they were talking about. It
Mm-hmm.
was hard to narrow down what type of chromatography experiment that was. Like, I
Yeah.
knew it had to do with affinity, but I thought that was too general statement,
Yeah.
which not it was not.
Yeah.
But I was like leaning somewhat towards one of these two, just knowing DNA was charged, and then again, then you brought out the fact that there was the positive charge from the nickel. So yeah,
Yeah. I was wrong though.
yeah,
So, like, that thing, I thought, like, the nickel basically, like, would uh like, dissociate, but it's just that weird thing, the nickel nitriloacetic acid agarose. Yeah, I should have listened to you when you said like agarose and stuff like that, because it's a very
all right.
specific biological thing, and histidine is what gets attached to it. So good job on that one. Yeah, I give that
Um,
to you.
I noticed when I was reviewing the Altius test, I finished reviewing it yesterday.
Mm-hmm.
I noticed a lot of the bio questions and even some of the chemical process, whatever it is. I noticed that um, I, for some reason, am getting a lot of the experimental questions wrong. Like if they're asking like, um, what would prove the hypothesis
Mm-hmm.
or control or whatever, like what could not be true, what could be true.
Yeah.
So I'm having a hard time getting through those.
Okay.
Um, I think this is kind of an example of one.
Yeah. Yeah.
Um,
All right. So, okay, let's check it out. So in the pre-incubation, so I see that in these, like, conditions.
yeah.
So let's see. So condition one, pre-incubated with biotinylated DNA substrate, after which non-biotinylated DNA substrate was added. Condition two, it was pre-incubated with non-biotinylated, after which biotinylated was added. And then three, it was pre-incubated with a buffer after both biotinylated and non-biotinylated were added. Okay. Then after substrate exposure, the reactions were initiated by the addition of like tritium labeled methyl donating substrate, which undergoes beta. Okay. Let's see. Then it was loaded onto plates that has something that binds to biotin, and then washed extensively prior to. Okay, so when they wash it, they're taking away the thing that doesn't bind to the biotin, it seems.
Yeah, I think I kind of figured that out. I don't really know what this cell wash or washing crap is. Like, I'm not a biochem person.
So it's just
Um,
like if in the it's like in the chromatography thing, like let's say the thing that we want elutes out, and like that chloride in that first example is still bound to like the positively charged stationary phase. So when we wash it, we like just get we get that sodium off. It's like.
Gotcha.
Mm-hmm.
Yeah, I saw a question like that from LTS yesterday too. It wasn't worded the same, but
Mm-hmm.
same thing. All right, so
Yeah.
they're
Now.
basically just asking after washing, what molecule are we removing, basically?
Yeah. So
So
I
that
want
would
to.
be whatever was left behind in the chromatography column.
Yeah, I want to take a look at this figure one thing though, because it says that
Okay.
um, a methyl donating substrate uh, gives off some type of whatever decay. Let's see. Wait. Okay. Sample or wash extensively prior to analysis by scintillation counting. Methylation rate. Um. Okay. Let's see. Biotin. So, what would bind to biotin? I'm trying to think. Oh, I mean, well, okay, that thing, but okay. So, if the thing is, so okay, the the avidin thing binds to biotin, right?
Okay.
And then they were washed. So the things that come out in the wash, like removed from the plate, are the things that don't bind to biotin, right?
Okay.
So we need to think which of these do not bind to biotin.
I would think two. I would think 2 and 3 because non-biotinylated substrate. If it wasn't biotinylated, then it would wash out. And then I'm thinking the methylated cytosine.
Yeah, now I need to see like what methylation has to do. Like, can you see that top? Let's see. So okay. All right. 30. Okay. So okay, let's see. So after they do the wash, they do like a scintillation count, which shows you like the whatever relative methylation rate.
Okay.
So condition one, if they used biotinylated substrate, there's a high methylation rate for the 509 okay, 509 mer, okay. Condition two, pre-incubated with non-biotinylated, it's like basically nothing. And all right, so if it's pre-incubated with biotinylated DNA substrate, there's high rate of methylation. So, okay, so then how does that connect to this? Let's see. So, All right, so we're trying to determine if it's going to be 2 only or 2 and 3 only, right?
So I was wondering something when you were going through that, like if it's non, if you put a non-biotinylated substrate in the column,
Mm-hmm.
why would you need to? If it's if it's non-biotinylated, I don't think it would bind to biotin. So why would you need to like wash that off?
Oh, because it. Wait.
So
Mm-hmm.
it's saying binding reaction designed to remove which molecules from the plate.
Mm-hmm.
Were we talking? Is this after we've gotten through the column? We're on the plate now, or is this
Um.
like
Yeah, I think so. I think that or we don't even have to worry about that column. Just think
I just don't know
about.
what it matters if non-biotinylated DNA substrate is added. It wouldn't bind to biotin.
Mm-hmm.
So I don't know why that would be true or why that like is applicable. But for number two, I mean, because like I know I think you would have to get rid of one through washing, and I assume three through washing, but I'm not sure about two.
Mm-hmm.
I might be like getting backwards now on what I was thinking because I was originally thinking it was two and three,
Mm-hmm.
but I just don't understand why you would need to remove non-biotinylated substrate if it can't bind to biotin.
So yeah, no. If it can't bind to biotin, it's like on a plate at this point, not like a column.
Okay, so we've alluded then. That's what you're saying.
Yeah, it's not going to do it
It's more hard to
automatically.
visualize that. Yeah,
So yeah,
because
it's
that's
like
the plate.
yeah, it's on a plate, and so we take the stuff that we have, which is all the Roman numeral stuff,
Okay.
and something's gonna bind to the biotin, and that's what we want, and what we wash away is what we don't want. So I'm trying to see if it's going to be the non-biotinylated substrate only, or is the methylated cytosine going to be a part of that? So that's why I was looking at the scintillation counting. Figure 1.
I wouldn't understand how to use that like figure. So I understand if you look at the 509 mer condition one versus the 30 mer, there's decreased methylation.
Mm-hmm.
So that's like the main difference I'm saying.
Yeah.
Um,
Wait, what was I mean? So the main thing that I see is that in condition 1, there's a crap ton of methylation. So that leads me to believe that, well, if we had washed away the methylated cytosine, maybe we wouldn't see such a high methylation rate. Because we're certain about the non-biotinylated part, right?
yeah, I'm getting like lost.
So we're certain about the non-biotinylated part, right?
Okay.
Like right?
Yeah.
Like that's what you said. It makes sense, right? Because
It did, and now I'm questioning everything.
oh, okay.
Actually,
So check it out. So if it's biotinylated, it already has biotin, right? So it's not going to bind to biotin again.
So I'm like confused now, like just
Mm-hmm.
understanding what the hell they're doing. Like I know they're trying to like wash this off to get what they want to get to, and I'm like forgetting and like missing what we're trying to isolate here.
Oh, sorry. Okay, so basically they're saying that um
Well, they're asking us that, like, what are we trying to get? And I'm just like, I don't understand what we're trying to get. Like,
mm-hmm.
yeah, I'm just like lost with what we're
Okay.
trying to. I guess they're
All
trying to
right.
steer. They're basically saying like that I need to figure out what they're trying to isolate based off
Okay.
what
All
tools
right.
we've given.
All right, check this out. So let's like take it from the top then. So
Okay.
what does this do, right? It methylates yeah, it
Cytosine.
methylates DNA, right?
Mm-hmm.
Okay. Now, do you know what they mean? So where do they say the 30 mer and the 509 mer? Those are nucleotides, right? Like
Okay.
nucleotide substrates, aka like parts
So
of
like your
DNA.
bases. All right.
Mm-hmm.
Yeah. Okay.
So so basically, you know, the DNMT thing, the presence of it would cause these nucleotide substrates to be methylated, right?
Okay. So in case of the 509er substrate, we're highly methylated versus the 30 we're not. Like a decreased methylation.
Yeah, it seems like in the condition of like whether it's whatever the 509 mer or 30 mer aren't really that important now because like the 30 mer is just 30 nucleotides, 509 mer is like the
Yeah, that's true.
509 ones. Um, so I'm just trying to look at because the conditions are the ones that say like biotinylated or not. Like condition one, right? It's pre-incubated with biotinylated DNA substrate. So the DNA, like the 509 mer, it has biotin on it.
Okay, so like I'm thinking right now we don't need to remove the non-biotinylated substrate again. So number two.
Yeah, yeah.
Okay, because that doesn't affect anything we're looking for since it doesn't bind to the whatever it's called. Where was it? The
Yeah, the
biotin. Yeah, so it doesn't. So two. So I would say probably two's out.
wait,
Um, right?
I thought you were saying the opposite there.
No. Wait, why would we need to remove the if we have like this stuff on our plate? I don't understand why you. I mean, I guess you could take the non-biotinylated out, but it's not interacting with anything.
Well, okay. So the plate binds to biotin, right?
Okay.
So when you wash it, so when something binds to biotin, it gets biotinylated. It's a weird word, but that's what it just means
I
that
kind of understand what it means,
yeah,
but
yeah. Okay, so if it's binding to biotin, right? Everything so it's biotinylated, right? And then when you wash it, everything that isn't bound to biotin, aka non-biotinylated, are just going to wash away.
okay. So then,
So you
so
have
the
the right idea first with non-biotinylated substrate. What I was trying to figure out was is like what about the methylated cytosine? And I
well
think
they use in the experiment. They were talking about methyl transfer. I don't know why they'd want to remove it, but
oh well they're not really removing it, right? Because these are like substrates. Like all of these choices are substrates, right? And the enzyme works on a substrate. Like, remember that term?
Yep.
So basically, what they're doing is, right, they're taking that. So, like, in all these conditions, they have the DNMT thing, which is an enzyme that methylates DNA, right?
Yep.
So, in condition one, they have the enzyme and they have it with biotinylated DNA.
Okay.
Yeah. As a pre-incubation step. And condition two, they have the enzyme with non-biotinylated DNA.
Okay.
All right. Now, in that chart there, or yeah, in the graph, it shows that in condition one, when there's biotinylated DNA, there's a lot of methylation, which means that that enzyme, the DNMT, is doing its job. It's working.
Okay.
All right. Now, since we do that scintillation counting, like the figure one, after we wash it, right?
Yep.
And we see that the methylation rate's high, then that leads me to think that whatever methylated cytosine is there is not washed away.
Okay, let me see if I understand this. So basically, we're looking at that. Basically, we're looking at conditions 1 through 3, as well as the table to kind of help with the methylation aspect of it.
Mm-hmm.
So we're saying, so the reactions, so the wash is designed to remove which molecules from the plate, and we're saying that we want to get rid of the not, since the plate's biotinylated, we want to remove what's not the non-biotinylated substrates off the plate.
Yeah, the plate has something that binds to biotin, right? So something that's biotinylated will bind to it, right?
Okay.
To the plate.
And I just, yeah.
So
And I see condition. I see, I mean, I didn't know what this meant, but I could still see the methylation rate is what they're talking about. So
yeah.
Condition 1 versus 2 and 3 had high methylation, and 2 and 3, it was reduced.
Mm-hmm.
Um,
Yeah. So overall, I just see that basically that methylation is happening. So if
if methylation's happening even like a little bit, then it's not like it's been completely stopped. There's still methylation.
Yeah, because think about it. If you washed away the methylated cytosine, you're not gonna see like any high amount of methylation, right? Like rate.
It would probably, I mean, I don't want to overthink this question
Sure,
going
sure.
back to some stuff up here, but like it would probably, if there wasn't any methylation, it would probably make it inactive. And I think that they use methylation in certain genetics. Like, I'm just thinking back, so I know methylation
Oh, like
stops something from
epigenetics.
happening. Yeah.
Yeah, I put
Yeah.
a thing in your notebook about that.
So I know they methylate things to like stop them from happening. I've seen like I can't think exactly, but like I think it has to do with DNA and stuff like that. But
Yeah, yeah.
there's
I have a thing in your notebook
like
about it.
gene stuff. Yeah.
In your notebook, it's called epigenetics. We can talk about that like after, like what the purpose of methylation is. But here we're just trying to see what we're removing from the plate, right? The plate binds biotin, so the plate is going to be bound to any biotinylated substrate. So when we wash it, the biotinylated substrate stays. The non-biotinylated substrate gets washed away.
Cool. Yeah.
So we're certain about that, but the methylated cytosine, what happens to that? I think that it's going to stay because of the methylation rates that we see happening.
Okay.
And since
Yeah,
methylated cytosine isn't binding to biotin. So that's why I would think it's 2 only.
okay. And like methylation's not completely gone because I assumed that would mess up stability or something. And
Mm-hmm.
yeah, okay. So probably two.
Yeah. All right. So they're saying that, yeah, the biotinylated substrate is bound to the avidin, right? That's the thing that binds biotin. And it's so tight, it's so strong of bond that the it's not going to be removed when you wash it.
Yep.
And then if it's non-biotinylated, it's not going to bind to it, and it'll be washed away.
Gotcha.
And then
Yeah.
then this is where we know about the methyl thing. The step after the wash is a scintillation count, which would be used to so it's just that figure. But if you wanted to know, scintillation is count is just a way to measure some type of radioactive decay. Like a Geiger counter, like when you go, it's like in a
Yeah, I
radioactive
know what that
area.
is. I forgot about it already.
Oh yeah, no, it's not not it's like just from movies and stuff, like like they're holding it in a radioactive area and it's like makes that sound.
Oh, I know what you're talking about then. Okay.
Yeah,
Yeah, yeah, yeah.
so that's just because it's because it says it undergoes beta decay, so it's just a radioactive particle that shoots out, and a scintillation counter, like a Geiger counter, counts the amount of like radioactive particles that hit it.
Gotcha. So like
Mm-hmm.
this was very difficult for me. Like I
No,
was
I
on the
think you
right
had the right
track.
idea. We just spent
Yeah.
our like we did. This is a good use of our time. We spent our time like talking about like you know like a lot of other topics like within this.
Yeah, for sure. And I just am frustrated because like
Mm-hmm.
you're timed on this test, and I don't see how kids are getting like high scores on this. With like
Just
my
so
biochem professor has a PhD and she's like an
uh
expert,
huh.
but like I don't know if she would get some of this stuff right.
Yeah, no, like don't think about time yet. Think about understanding every single bit of it. Like think about like you said you'd study like exercise like science, like
Yeah,
foam rolling. Do you know what I mean?
yeah,
Like or
yeah,
like deep myofascial relax, like that type of stuff, deep tissue massage.
yeah.
That's like what we're doing here. We're doing like we're going like we're really like exercising our brain here to basically like because it's not about just getting as many questions. Like people can do this and answer questions. Then like I think I told you about this. Like people, a lot of people like they just answer as many questions as possible, and then they look at the ones they got wrong, and they look at the answer, and then they tell themselves, "All right, I get it now. I'm not going to get it wrong next time." But they're not going to ask that same question again the next time.
That's
You
true.
want to
Yeah,
like have a real flexible brain, and you're doing that. Like what you're saying with like substrate stationary phase and like all the stuff you've been saying.
yeah.
Excellent. Okay, so yeah, so okay, let's see. Which statement about the interaction between that and the DNA substrates, and also like since physics is so like small on this test and in medical career, and like that's what I majored in. So like, how is any of this working, right? And it's just because, you know, it's like you spend more time on understanding like the conceptual things, as long as it
Yeah, the
takes.
AMC hates us all who did not pick a hard science major like math or physics, I guess.
Which is weird because it doesn't like test like that. But you could see the same thing for every single test. Just Google like LSAT scores by major, GMAT scores by major. Every single test, you'll see that same pattern. And how is that possible? Like physics isn't on any of those tests, right? And it just it's like exercising your brain. Think of it that way, okay?
We'll do it.
Like because you think of because you studied exercise, right? So like by the time you do take the test, it's going to be internalized and like muscle memory, but brain memory. but like, you know what I mean?
Yeah, for sure.
Okay. All right. So here we go. So which statement about the interaction between this and DNA substrates is supported by the data in Figure 1? Okay. So in Figure 1, it seems like the okay, so let's see. Let's just go by the choices. Biotinylation prevents DNA strands from binding. So it seems
That
like
couldn't be true, I don't think, because we're still having methylation.
Mhm.
So I think
um
you can go on how in depth you want to go with it. I would assume I'm probably trying to use constant knowledge here again, but like I know
uh-huh.
if I think there's something about if you methylate DNA, something like it prevents like gene
Yeah.
yeah
You're talking about the epigenetics thing.
yeah.
So we can talk about that, but for the purposes of this, we're just looking at like what we can like which of these statements would be true in fit like Figure 1. Like we're not we're more concerned about the enzyme and how it works than we are about the purpose behind, you know, methylating DNA and having, you know, RNA being expressed at a lower rate, which is what you're saying, right?
Okay. I just don't. I mean, I don't think that could be true because based off what we know up here and then looking here and seeing that methylation is still occurring.
Yeah. The biggest difference that I see in Figure 1 is just between the 509 mer and the 30 mer. Right?
Yeah.
That's like the first thing, you know, right?
Okay.
So I would think that so B and C seem to be talking about what I'm noticing.
It says stability of the protein DNA fibers formed on 30-mer and 5-9-mer DNA strands was the same. I mean, it doesn't appear the same to me.
Doesn't appear the same to me either.
Yeah. So I would strike that out just because they're not
Yeah.
the same. Stability of the protein DNA fibers formed on the 5-9-mer strand. Well, if there's increased methylation, I assume there's going to be more stability. So that could probably be true.
I would go.
Biotinylated DNA strands are methylated at a higher rate than non-biotinylated. I don't think that the graph was saying they talked about the rate of methylation, but
Yeah,
the
so
graph itself didn't show whether it didn't. Oh wait, it does say conditions. Hold on, it does say conditions. So
so the conditions, yeah, but they're talking about like the scintillation thing happened after washing, right?
oh yeah, okay.
So that means we got rid of the non biotinylated DNAs, right? So we're only dealing with biotinylated DNA. So D, it's talking about not like a higher rate than non biotinylated DNA. We're not dealing with non biotinylated DNA anymore. We washed that out. Right?
Okay, yeah, I didn't pick that up right away. I would have like gone back and looked.
Mm-hmm.
Yeah, so I guess
And then yeah,
I
and
wasn't
then
remembering that. Yeah,
oh yeah. And then A
so
says biotinylation prevents DNA from binding to it. We're only dealing with biotinylated DNA now, right? And if it being biotinylated prevents it from binding to the enzyme, then we wouldn't have any methylation happen,
okay,
right?
yeah,
So so like right away C seemed to be the right answer to you, and that's good. But now we
yeah,
know it's the right answer because of what we know about that washing step. So
yeah. So sometimes this happens to me, like when I'm doing it on my own. Like I start second guessing myself, and like I'm just not like I would have been right here, and then I would have wasted time. So I would have gone to that, and I probably would have gone back to the red stuff again because I would have not remembered.
oh yeah, no. So like don't worry about like how it would be on the actual test. On the actual test, yeah, you'd hit C and you'd move on.
Yeah.
Since we're not taking an actual test now, we're basically foam rolling, and we're basically knowing not just that C is right, but why every other answer is wrong.
Okay, I'm still a little lost with D, like because I looked back and I was like pretty sure when I read that I was like that's probably the answer. Let me check this, and then I'm like, oh wait, well it's showing the 509mer is highly methylated,
Mm-hmm.
and there might be a difference, but I felt like this was probably a trick
It
question, not
is.
necessarily, yeah, because like I was mainly looking at it from the purpose of
I'll
they were
tell you.
saying this was
I'll tell
just
you why.
comparing 509mer
Yeah,
versus
I'll tell you why it's a trick question. Because
okay.
you see condition one has the highest methylation rate,
Mm-hmm.
and then look up where it talks about condition one, right? Like up, you know.
So right, so actually in condition one,
Yeah, it says
or
that it
look
was
up here.
pre-incubated with biotinylated DNA, right?
After which non-biotinylated DNA was added.
Yeah. Now look at condition two. It says non-biotinylated DNA, right?
Okay.
So that is what makes. That's why it's like a trick, because condition one looks like it's biotinylated DNA. Condition two looks like it's non-biotinylated DNA, right? And figure one shows condition one has a higher methylation rate. Condition two has a lower methylation rate. So that could lead someone to believe that biotinylated DNA gets methylated at a higher rate, right?
Mm-hmm.
Like that
Yeah,
would say,
that would have had me stuck. Like I'm confused now a little bit still. Like
mm-hmm.
that would trip me up and make me still not sure. So
So the reason that it's not going to be that is because if you go back to that previous question, so what so we do that, we get the data for figure 1 after we wash it, right?
yeah,
And when we wash it, what gets removed?
the non-biotinylated substrate.
So an example of that is non-biotinylated DNA substrate, right?
Okay.
So that's gone completely. So everything that you see there is biotinylated DNA in figure 1. See how it says washed prior to?
We're
Oh, where you were highlighting right there?
here. Oh, I see it now. I keep not seeing that samples were loaded on displays containing what was bit of events washed extensively prior. So the results were done. I'm getting that confused though. So it was prior to. Hold on.
Mm-hmm.
So they washed.
Mm-hmm.
Okay, so they washed everything before they got the
Yeah.
methylation results.
Mm-hmm.
Okay, I'm following that. So basically, methylation, biotinylated versus non-biotinylated doesn't matter because we washed it before the results, and that hold on, we
Mm-hmm.
washed it before we got the results. So therefore, it doesn't matter because the non-biotinylated is gone. It's just this is just showing biotinylated
Yes.
methylation. I
Yes.
got that now. I still probably would chose C,
You
but
would have
that if
chose
I had
C.
gone back and like went through too thoroughly, like that would have possibly tripped me up.
Yeah, so this is like the importance of knowing like how, like when I look at D, right? I see how people can choose that. And then that gives you insight into the test maker, the test creators, how they make the questions.
Yeah.
Because if you just looked at condition 1 having a higher methylation rate than condition 2, and you looked up and saw that condition 1 said it was pre-incubated with biotinylated DNA, and condition 2 says pre-incubated with non-biotinylated DNA, that would make you think that biotinylated DNA has a higher methylation rate. But that's not the answer because of that washing step. So...
But I feel like that partially doesn't answer their question very well either, though, just because they were talking about the interaction, which to me, I'm thinking of the binding interaction and stability anyway, automatically as I'm reading this, and this kind of talks about that.
Yeah.
This was a more specific answer.
Yeah.
Even reading it, it obviously made sense.
Mm-hmm.
I could see how you could want to, like, pick this answer, though.
Yeah.
This kind of happens to me occasionally. Like, I'm sure about one answer, then I'll switch it because I'll see something. But, like, I see that was a trick question. So, yeah.
Yeah,
that was a tough one. I mean, it wasn't that tough. Like, I think I had it right. I had it.
Yeah, you definitely had it. I just used this as an opportunity to, like, show you how they to get into the mind of the people who make the test.
Sometimes I notice that I'm usually pretty good at eliminating 2 wrong answers, but I'll get stuck like that and things will seem like they're true, and I have a hard time completely, like, confidently answering and moving on.
Yeah, because listen, this is how they make tests, right? They basically they have people take, like, a sample, right? They see what people get right and wrong, and things that they get wrong, they make choices that are like that. Because if the answer was just, like, blaringly obvious, then everyone's going to get the same score, right?
Yeah, no, it worked for me in undergrad where I just memorized everything.
Exactly.
Just verbatim.
Mm-hmm. So these are, that's what people did, and now you're taking a test that is meant to distinguish you from those people who also did that.
Yeah, I feel like I'm also being punished by the AMC for, like, ending all my notes and, like, memorizing everything. Yeah, it sucks. That's helpful going through it, though, because, like, I feel like I'm really close, even though I've done pretty poorly. Like, I'm really close, but it's just, like, I'm picking the wrong answer still.
Yeah. So, yeah, that's why, we're going through, like that, like, again, like, when I say, like, when people, like, when they do, like, a bunch of questions, they look at what they got wrong, they look at the answer, and they say, I'm not going to get it wrong again. Instead of doing that, we're basically, like, I'm basically forcing you to not only tell me why the right answer is right, but why the wrong answers are wrong.
For sure. I mean, I've done
a complete understanding. Yeah. Then you have a complete, like, it's foam rolling.
Yeah. Can we do like one CARS passage? I've done these a long time ago. Let me try to make sure I find one that I don't remember.
Okay.
I don't know if it'll be helpful or not, but I can show you, like, what I did. I did the new one. Where was it at? Hold on. I think it was Q.
We could just do like a passage, like, out of.
Oh, here it is. I was going to show you, like, my diagnostic. I did all these, and I did it kind of, like, untimed. But I was going to try to show you, like, what I'm getting.
Yeah.
So, like, I finished this one at 73% correct.
Okay, this is useful. So, So it seems like the type of questions you're getting are pretty much similar. But a lot of this comes down to the type of passages that people have trouble with.
Yeah, and I noticed I tend to
it seems like you did well on, like, all of these.
Except, well, these I did pretty good. This is the most recent CARS stuff. I think the hardest I've had trouble, not this one, maybe the other one would show. I do notice I do better on things that I enjoy.
It's funny, they spelled archaeology wrong.
Yeah. Yeah.
I'm really fast at noticing.
I tend to do better foundation-wise across the board on all my sections. I do better at comprehension or, like, general content than I do with, like, the critical thinking, analysis, reasoning stuff.
Yeah. So foundations, that's, like, main idea. Beyond the text is inference. Within the text is just grunt work where you just look at it.
Yeah, like this one I finished. I did really bad on that Picasso one. Yeah, so like it's pretty much the same pattern
Mm-hmm.
here. I did really bad on one passage, but like
So I see what I
I was
see
probably
for
higher.
lots of students, like art history and philosophy. That is usually what trips up. Those types of passages usually trip up lots of
Yeah, I have
students.
a hard time with the
Mm-hmm.
philosophy type passages the most. I think I've gotten better at them.
Yeah.
It's just harder to read because I don't find it very interesting.
Yeah,
Like,
yeah, yeah. And
um,
it's like written it's not written in like it's written in a much more denser form.
so as far as like we can go through it if you want to read out loud or whatever. Let me find one I don't remember. Just try to
Mm-hmm.
you just do it maybe one or two. Trying to get to the middle somewhere. Can't remember that one. I mean, I'll probably won't remember the answers. It's been close to a year since I've done these, but
Mm-hmm.
maybe I can start with this one. So, like, basically, I've tried a million and one like strategies to go through cars,
Yeah.
and I find what's happening is sometimes I don't comprehend it as well as I could, and then I
Yeah.
get to the questions. I feel like I have to look back too much,
Yeah.
and then I waste a lot of time between two answers. So what I usually do is I'll take about 4 to 5 minutes to read through the passage,
Yeah.
and usually I'll try to highlight keywords or things
Yeah.
that seem important that I might have to go back to because it helps me to remember it.
Okay.
Um, and
So
then
yeah.
then I'll go through and kind of like
Okay.
go back. I
So
tried like writing an outline that didn't work for
no,
me. I tried.
that's
Uh,
so let's do
yeah.
this. So basically like
Okay.
we're going to go like paragraph by paragraph, and I'm going to ask you like basically what the point is, like what the and it's just like a word that you need to know for the point. Like it doesn't have to be like anything like too um like extensive,
Okay.
and also be aware of like generally speaking like where things are in the passage, like where things are talked about. And so we're gonna start like slow first, like I'm gonna like actually read it like
Okay.
out loud, and if there's any like word you don't know, uh, ask me.
Okay.
Um, okay. Um, so all right, ready?
Yep.
Okay. No matter how noble the effort, the burden of proof always lies with the reformer. Many empirically sound proposals to increase the effectiveness of elementary schools in the United States have been dismissed with the response, if it is so necessary, why has the need not been recognized before? To counter this response, a reformer should make clear that a problem has been identified. So,
Okay.
yeah. So what should be?
So
Mm-hmm.
there's kind of a few things there, but they were basically um, the author's tone was kind of like dismissing the way they've been addressing increasing like effectiveness of public schools. Um, I don't know what the problem is yet. Maybe they haven't gotten to that yet. Um,
Oh, okay.
they're talking about the concept of the reformer.
Yeah, so exactly. So it's less to do with so cars. It's like separating the wheat from the chaff, like separating like and the important stuff is like almost like 5% of like the total words there. So like um so paragraph 1, all that really matters here is that so do you know what they mean by the burden of proof lies with the reformer?
Um, Vaguely, it's a burden of proof. I'm not really sure exactly what they mean by that, but I think they're saying basically. I think he's saying that what a reformer should do
Mm-hmm. Yeah.
is identify what the problem is and
Yeah.
yes, burden of proof.
Mm-hmm.
He talked about he's basically just saying no matter
Mm-hmm.
how much effort you put into whatever the change might be or whatnot.
So all right, let me so burden of proof, right? Let me give you like this example, right? It's like a common one by this philosopher like named Burton Russell. He says All right, let me tell. If I tell you this, there's a teapot flying out in outer space, and then you're gonna tell me, like, what are you talking about? Like, what are you talking about? Like, can you prove that, right? And then I tell you, well, you can't prove that there isn't. So the thing here is that if someone is making an extraordinary claim, like there's a teapot out flying in space, the burden of proof is on that person who's making that outrageous claim. They can't say something outrageous and tell the other person, prove me wrong. The burden of proof means that the person has to prove themselves right, not the other person.
Okay. Yeah.
So
that makes
that's
sense.
what the burden of proof means. So, like, okay. So they're saying for a reformer, right? If they want to reform school or something, whatever, it doesn't matter what it is, the reformer has the burden of proof. The reformer can't say anything without the reformer can't just say, we need to make schools better, right? The reformer has to say, there is a problem. Here is the proof that there is a problem. Now we can start to talk about it. So, I hope that kind of made sense.
Yeah, that made sense. And I've like in terms of highlighting,
Oh yeah, I would
like
say
should I highlight or not? Like I probably, if I had done it, I probably would have highlighted.
I would highlight like not like entire sentences but like words, and you can do that by double clicking on it and then like hitting like Alt H or whatever, or like whatever it is for Mac.
Yeah, I
So
have a
I would
Mac. I don't know what it is,
so
but
I would
yeah. So
so click on burden.
that was up here.
Yeah.
I probably would have. Yeah, like burden. I probably done burden of proof. So just burden.
And then proof. And then reformer.
Reformer.
Yeah. Pretty much.
But isn't this kind of the thesis though? A reformer should make clear that a problem has been identified, or no? Or is that kind of the?
It's an example of what's there up there.
Yeah. So I probably don't need to highlight anything else. That's probably all I needed.
I would say so. That's
Okay.
that's how I would do it. Basically, the since you understand it, right? Like, so all you need to know is that the burden of proof is on the reformer. If there is anything wrong, like let's say you want to do prison reform, right? You can't just say we need to do prison reform. You need to identify the problem. Like if you want to reform something as a reformer, you need to like be the one to show proof that something's wrong in the first place.
Okay.
Okay.
Cool.
So yeah. So basically that's that for that. Oh wait, where are we? Oh yeah, go up. Oh, this is the next paragraph. Okay. All right. If the condition addressed has not been completely and clearly established as a problem, those concerned should ensure that it is accurately measured. The appropriate instrument for measuring educational effectiveness is a test noted for its reliability and validity. That's like psych-social stuff, right?
Yep.
If the researchers believe that no existing test is adequate, they should develop their own test. Since the burden of proof for their methods is focused on their instrument, sincere reformers will be very serious about establishing its credentials. All right, what is that saying?
If you're serious at addressing your problem, um, you have to make sure like your instruments are valid and reliable, or you're not gonna have any credentials. It's not ever gonna happen.
Yeah, you're gonna need to have a good like measuring system to show show that there's that problem, right? Like if I say I want to reform prisons, there's a problem. The problem is they're crowded. Sorry. The problem is that they're crowded. then then it's like, how do you know they're crowded? Then you have to be like, okay, according to this, like, whatever, like I don't know, like study of like number of people that are incarcerated, number of jail cells that are available. Like basically, it's just showing you like that you need like a good high standard of evidence when you present your case.
For sure.
All right.
I mean, I might have not highlighted, but if I did, I
I don't
probably would
think.
have just,
Yeah, I think you get it.
yeah, I was just thinking like, sincere formers use reliable and valid tests. So I probably maybe would have highlighted that in those two words, but
Yeah, I think. Yeah, I think we're good because they're talking about like, um, you know, like in theory, like what a reformer should do. So we don't have to be too like we don't have to be too like held back by any specifics.
okay.
Okay. Now, when a proposed intervention is not justified in the most minimal fashion, the public has to wonder why not. It is thus reasonable to be suspicious of the promoters of the Generalized School Readiness Program. What is their motivation? Are they agents of an unfriendly power bent on dumbing down U.S. education? Are educational entrepreneurs trying cynically to profit from the general dissatisfaction with the nation's schools?
Um, so the author is just giving examples and asking questions like relating to
So here,
promoting like the current education system as it is.
so here I would highlight something, but again,
Okay.
I'm separate like the statements from the like evidence, right? Because they're gonna so like the reasoning within the text questions would be like, what does the author like? What does the author use to support this statement, right? That's when you would go back and look at like the details, like um, the school readiness program. That's an example. So like separate the examples from like the actual like points or arguments that are being made. So here the argument that's made, right, is like, so when the reformer, right, says, all right, this is how we're going to fix this, right? People, the general public, expect the fix to be very, very tiny. They don't want like a massive overhaul. Because if it's a massive overhaul, they're going to wonder why. Why is it a massive overhaul? Why isn't it just a tiny little like thing? If it's a massive overhaul, they're going to be like resistant to it. They're going to be like, are you like, trying to completely change America? Or are you trying to completely change, like, do you not believe, like, blah 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 blah. So here the thing I would like highlight is intervention. um Minimal. I don't know, like, what, like, basically that's just saying that the intervention, they want it to be as minimal as possible.
Gotcha. Yeah, sometimes with these, like, where they cite maybe two examples, I have a hard time highlighting wherever their point is. Like, I kind of was following it, but yeah, sometimes that's where I excessively would highlight. Like,
Yeah.
I mean, I see they're asking questions like he's trying to get down to why are they doing this, and
those are just examples of the of if there's if the intervention is not minimal, then they're going to ask, like, they're going to wonder these things, and those are examples of things that they might wonder.
okay,
But overall, what we're getting here, like what we've been highlighting so far, is like what the reformer should do, right?
yeah,
Okay. So far we know that the reformer needs to identify a problem, the reformer needs to provide really good evidence for the problem, like using, measurements. And if the reformer presents a solution, the solution should be like minimal. Like it shouldn't be like this a massive overhaul kind of thing. Is that What do you think?
Yeah, I feel like I'm following.
Okay.
Sometimes when I read CARS passages, just time constraint reasons, like
Yeah.
sometimes I won't fully absorb everything. Like I usually have a generalized idea, but sometimes like I just have to keep reading and hope that it makes sense altogether when I come through the whole passage.
Oh yeah, yeah. So
The way
far,
we've been doing it, I understand it pretty
yeah,
well.
because you
I
see,
like this. I wouldn't have known exact. I kind of had the gist of what they were saying, but I didn't
yeah,
have it like as well as how we had
yeah.
talked
So
about
you
it.
see how we're basically like just we're see the author's point, and then the things that come after are examples that the author is using to advance or strengthen that point or argument. So we're taking we're really like putting this passage on a diet, we're getting like the actual like point. So we don't have to be bogged down by any particular details. Okay, so such speculations may appear to border on the absurd. By the way, by speculations, they're saying the things that the public might think or wonder if your intervention is not minimal. Okay, so such speculations may appear to border on the absurd. However, stranger motivations have been discovered. It's more useful, however, to assume that the promoters, wishing to keep their business financially solvent, have opted not to address school-based problems from the viewpoint of children or parents or even teachers. They are merely following the usual practice of blah, 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 blah. That's just...
They're just saying it's basically like just corporate like BS. It's not helping the learner.
they're just so they're taking the example that we don't really care about, and they're talking more about that. So that's more like kind of like fluff that only comes into relevance when they ask us about it.
I noticed up here, like they said, speculations. The author said it may appear absurd, but then he isn't contradicting himself, but like he's saying there's stranger motivations
Yeah.
in like what was speculated. Is that what he's saying? Like I probably would have highlighted some of that, but not that I have to. Like I understand it.
Yeah,
I guess
yeah.
like I don't know why. I just feel like I'm gonna if I don't highlight, I just get like sketched out that like I'm gonna like not know what to look back on. I think I probably use it as a crush that wastes more time than helps. Probably.
Yeah,
Well,
yeah.
I'm gonna let the dog out. They're probably barking at thunder.
At what?
They're probably barking at like the thunder and everything.
Okay. So yeah. So you get the idea. So yeah, you don't have to worry about like excessive highlighting or anything like that. Okay. So all right, then wait. Piaget and Gesell or whatever from different schools of thought also had direct experience with children in an educational setting, and both contributed profoundly useful principles to the field of education. Yet the conclusions of both about the need to consider developmental level are opposed by advocates of generalized school readiness. So they're really going off on this particular example. So expect questions about this. But that's just basically saying these are so-called experts in the field of education, right? But this intervention thing that they're talking about. So now we've established the firm example that we're using for intervention, and it's that generalized school readiness thing. So keep that in mind, right?
Okay.
So because now they're using counter examples to say this intervention strategy has whatever experts that are against it and stuff. Okay, next one must wonder about the experience these self-proclaimed experts have had with children. Also, like, keep in mind, like, tone, like, self-proclaimed.
Yeah, he's pretty harsh. He's
Got it.
pretty
You
like,
got it. All right.
yeah.
Good. You have a pretty good instinct for this. Okay. Their description of a child learning to draw, for example, assumes a struggle from stage to stage. Most modern observers of children think that if a task is developmentally appropriate and has personal meaning for a child, it is approached as a pleasing challenge, not a struggle. Okay, so that's just, you get it. In the literature promoting their approach, the advocates of generalized readiness are clearly directing their appeal to school administrators. Parents who do not understand the readiness concept are dismissed as uncaring. All right, so overall, it seems like this guy is. So he talks about like what a reformer should be doing, right? And he's using the generalized school readiness thing as an example of what reformers are doing wrong.
Yep.
So I usually don't say this, but I think we can like check out the questions and then like look at the passage if we need to. Because I think you get the idea, and I think that the questions. I guess I. It seems like the all right, whatever. I'll just for completion's sake. Let's see. Literature promoting their pro. Oh no, sorry, I already said that. So people who don't understand this generalized whatever readiness concept are dismissed as uncaring, right?
Yeah.
Teachers who question it are described as uninitiated. So basically, they're trying to say that these reformers for this generalized school readiness program are really dismissive of people who question them. Because, the author thinks that these advocates of generalized school readiness need to be the ones that convince us. Instead of saying, trust us, we're right, and if you don't believe it, like, F you. Right? So,
yeah.
Okay, and then just last paragraph. Comparative data of alternative, okay. Poorly researched, okay. But slickly presented program, okay. Okay, so this one is basically kind of saying that something that's poorly researched but it's well presented, like the generalized school readiness program, that is like good because it's easy to sell. But if something that's better, like developmentally appropriate instruction, which parents are more likely to want to implement, it's hard to sell to decision makers concerned with uniformity. So, in the long run, however, it is the forgotten parents and children themselves that will pay for the short-sighted ambition of this policy. So, he's saying at the
He's
long...
pretty much dismissive of all the way that education is, all the programs they've tried, and there's not research to support it. And then he's basically kind of criticizing the parents too for not taking on the approach because it's more difficult to take on. It's more hands-on. It's not
he's
an easy way out.
Well, he's only criticizing the generalized school readiness kind of approach.
He's just saying they'll suffer the consequences.
Yeah, because it says developmentally appropriate instruction, which is different from the generalized school readiness thing. Parents are more likely to judge that to be reasonable, right? But it's just hard to sell. So
okay.
So it's basically the author is basically like saying that these reformers are not doing their job. They're just doing like something easy that they can benefit from because it's easy to sell, but at the end they're not really doing any like good they're not really reformers. They're not doing any good reformation. So I think you pretty much get it. So let's see. The author apparently considers a theory of education that has little to do with the learner. So okay, there's a quote there, so we're gonna look for it in the passage, reasoning within the passage here. So we're gonna look for basically where so it's probably gonna be where they talk about. Okay, there we go.
Right.
You see it, right?
No, I was looking at the questions that just happened to slide up.
Okay.
But like when I was,
we're gonna practice this together also, like as we work together, like
okay.
Command F in your brain. Do you know what I mean?
Yeah, I know what you're saying.
So show me where it says learner. Little to do with a learner. It's pretty much. So first of all, like it says theory of education. So look for where they talk about, like Piaget and those people.
Okay. I would have had it gone up and then went down, so.
All right, good. But they also talk about them in another paragraph, right? Maybe they stopped talking about them. Maybe before that. Mm-hmm.
Okay.
Do you see it?
No, I'm still looking at CPJ. I have a hard time hunting this crap down.
All right, it's okay. We're working on it.
Little to do with the learner. It's harder. It's easier when I'm doing it by myself, but we're kind of talking. It has little to do with the learner. Theories of education. Oh, here we go. So.
Yes.
Okay.
All right. So we found it. So now let's see let's try to see what the author feels, like how the author feels about it, right? So basically they're saying, that, these reformers and stuff, they don't really care about the viewpoint of the children or parents or teachers. They're just following the usual practice at the professional level, treating learning as an abstraction that has little to do with the learner, right?
Yeah, I felt good.
And then after that, he says what? He says that all these other people with empirical evidence, right? Like empirical evidence?
Yeah.
Do you see that?
Was that up here?
Well,
I remember reading it in the passage. There's somewhere about empirical evidence.
So it'll be like right.
Oh, I see it at the bottom. Never mind. I thought I saw it twice. I thought I saw it earlier. So the outlook.
So okay, first of all, how does the author feel about empirical evidence?
Children's intellectual all work to counter.
So before, all right. So check this out. So empirical evidence. How do you know what is meant by that? Empirical evidence?
Yeah, it's like you need it's just like research. You need like empirically like valid data information.
So how does the author feel about that? Does he like it?
I have a hard time figuring that out.
Well, I don't remember what he said about what a reformer needs in
he said they need empirical evidence to support what they're doing. So I was just trying to understand in the context of this sentence.
Oh, okay. So like quickly. So the author likes empirical evidence or no?
So I guess is he using them to support that he believes that because he's citing Piaget because I know I'm not trying to use outside context here, but he's saying these people use empirical evidence to support intellectual development.
So wait, quickly. So author, is he for empirical evidence or not?
I'd say he's for empirical evidence.
Good. So they're saying that the theory of education that has little to do with the learner, right? There's empirical evidence, right, that worked to counter that type of outlook.
Yeah. Okay.
So if he likes empirical evidence, right, and the empirical evidence is to counter the outlook that learning has little to do with the learner, he's going to not like that outlook.
So should I go through the questions now? Or do you want to solve?
Oh, okay. No, I was just like saying like yeah, that's how like the author like feels about it, right?
Okay, yeah.
So okay, so now what do we think about?
All right, so author apparently considers a
Mm-hmm.
theory of education that has little to do with the learner, practical but unethical. He never talks about it being unethical, as far as I understand. Um, we did talk in the passage about that it was basically easier to implement.
Okay.
So I'm not 100% sure I'm ruling that out yet.
Mm-hmm.
I don't think it was. I don't think we talked. I don't think he directly said it was unethical, unless I missed that. Um, Ethical, but not generally accepted. Well, he said it was. It is accepted by the public, so I would
Mm-hmm.
eliminate that. Generally accepted, but ineffective. I know it's generally accepted.
All right.
Um, it's not effective because it leaves learners behind, so that's possibly true.
Mm-hmm.
Effective, but impractical. It's well, this isn't effective. He would be against that. So I don't think A is true. I think it's probably C. Generally accepted, but ineffective. Um,
So now since practical and generally accepted might be kind of similar, let's think about ineffective versus unethical.
okay, so ineffective just means the
And don't worry about
I'll
like time, like it's okay,
yeah,
like we're
yeah.
gonna okay.
The so I know for sure it's generally accepted, but it ineffective would mean that it just doesn't work at all with like as far as like the learner goes, and as far as unethical goes, that would be saying that it harms the students potentially, which I guess towards the end we talked about it was kind of a short-sighted approach and the children themselves would pay, but I don't know that it was necessarily an unethical. I don't remember talking about anything in the passage in terms of ethics, like whether it was ethical, unethical. I mean, he was against the way basically like education system is continuing like the generalized school readiness program when they could do better.
Mm-hmm. I think
This is a good example of where I would pretty quickly do this and I'm like crap, like and
well
trying to like go back and support myself.
yeah, this is a good example of you like though noticing where the unethical part comes in, like at the end, right? So that kind of supports that. Where can we find support for the ineffective or effective part? See if if you see anything like that, right?
He talks about motivations.
Well, okay, so maybe how about this? Practical versus generally accepted. So this is where it's like 50-50 and you turned it into 51-49. And so like you may have the right answer, but I want you to like
Well,
be
the reason I would go with C for sure, like I
mm-hmm.
probably am like I'm like 90% sure I would pick C, is
Yeah, yeah.
because I just feel like unethical wasn't the he never directly like said aside from like that it potentially harms the children. I guess you could say it was unethical. I mean, all
Yeah,
right, let me
yeah.
go back to practical.
Try,
So
so what's practical versus generally accepted? Tell me like
I was thinking generally accepted because it said in the end, it said like basically in two incidences here that people
mm-hmm.
basically accepted
Yep.
this style of education, even though it doesn't have to do with even though it's not increasing learning per se,
Mm-hmm.
It's easy to do. It appeals to the parents, so they're just going to accept it, even if it's not the best way to do it. So I feel like he basically summed that up here.
Mm-hmm.
So I'd probably pick C. I just don't feel right about A. I
Uh-huh.
don't know that I mean, it is maybe more practical to do the easier way.
Yeah, but there is a difference between practical, right, and generally accepted, right?
Let me look at what he said down here again. This is probably where I would have gone. Uh, that's just kind of like his tone. Yeah, so this is where like right now I would kind of like I would have to pick it and move on, and like I'm not sure what I could do differently right now to
Mm-hmm.
to to find. Maybe I could go back to the this and revisit wherever that was. I think it was that paragraph 4. Yeah. I mean, they're saying here that basically he agrees with Piaget and Giselle's learning methods, and even knowing what we know about them, the general high school readiness program is basically against their developmental like style.
Mm-hmm.
I just don't feel like it's unethical necessarily. Like,
Uh-huh. So okay. The thing about unethical is true because of what he says at the end. But the thing I wanted you to focus on was practical versus
practical.
generally accepted. Just because
Well, I
some
know
people.
what practical means. I'm just having a hard time using it in terms of like,
Oh no no no! Of course of course.
yeah, yeah, yeah.
I'm not saying you. I'm
Oh
saying
no,
that
I know what you're saying. It's just like
there's like a
education a little bit
nuanced
more.
difference between the two. Like just
Yeah,
because. So like yeah. So like you know like something that's practical like still means like it's easier to like implement and stuff, right?
yeah. So I mean, I see that as being true too, though. Like, I feel like both are sort of true. Like, they did say he did say directly, basically that that was more practical, but there would be it wasn't the best way of learning, which is why I'm like torn because I feel like he's saying it's practical but ineffective, but he's also saying it was
So
he was
so
saying it was basically generally accepted by the parents and the school accept that.
okay. So we don't really know if it's practical because if it was practical, right, it would be easy to implement, right? And if it was easy to implement, remember, like minimal intervention, then people wouldn't
Yeah.
be like, up in arms about it, right? But it is generally accepted. And generally accepted doesn't mean that it's necessarily practical. It just means that more people um, accept it.
And I will be honest, I'm not 100% sure if he said it was ineffective. Um,
I think the
I think he
ineffective
kind of talked
part,
about the Piaget. He
yeah,
talks about them. Yeah.
yep,
So that's kind of where I would go with that.
yep,
All right.
yep. So, so yeah. Now, like, I just want your 90% certainty to go to like, kind of like 99 or 100. So, do you feel more confident that it's C now?
Yeah, I feel like it's C. I just wasn't sure about the ineffective until I read that part about the Piaget because I'd
Mm-hmm.
forgotten that. So I feel like I'm pretty much 100%. I don't think unethical had it. I would have rolled it out right away just because of that. But then I
But
know
you
I
did
needed to
see,
fact check about practical.
yeah, but you did see yourself, like you went right down to where they talk about it being unethical, like right
Yeah,
at the end. So
yeah.
unethical is in.
Oh
It's
yeah, that's right.
just practical. The verdict's out on practical. So basically, we have choice A where we don't know about practical, but we know about unethical, and choice C where we know about generally accepted and we know about ineffective. So now you now it must be C, right?
Yeah. Okay, so I was actually right about that. The part about the
Exactly.
unethics.
And you went there
Um,
immediately.
yeah. So yeah, this is just an example of like when I'm taking a test, I'm like crap. This
Mm-hmm.
happens to me like say like 2 to 5. If there's 5 questions, this would happen on like 2 of them, and sometimes I will pick the wrong one just because I'm like in
Mm-hmm.
a jam. So that was helpful to go through. But yeah, that's basically my biggest frustration with cards. Like
Mm-hmm.
if I could be more confident with my choice when I'm down between two, that I wouldn't miss hardly any.
Yeah.
Because
So
the only one I miss cars, it's this
yeah.
situation right here every time.
So I hope like it helped like looking at it like practical. There's a level of uncertainty there. Unethical, you're certain about that being right. And then generally accepted, you're certain about that being right. Ineffective, you're certain about that being right. So yeah, you're left with something that's 50%, like you're certain that it's right, and something that's 100% you're certain that it's right.
Yeah.
And in that
And I know
case, you
those two
choose
things
them.
were true, and the other example only had one that was
Yeah.
for true. It was the practical.
Mm-hmm.
Okay, cool.
Yeah. All right. Good. Okay. So the author argues that the reason for the approach taken by the promoters of the Generalized School Readiness Program is their wish to
All right, so that's where I would have gone to. We talked about the promoters and the generalized school readiness program.
yeah. So that's where
Excuse me.
like that's where the questions come in, like the suspicion, right? Now, what could be like one of the reasons? Like, what can be one of the actual like?
So why the promote? So he's saying why would the promoters promote? Why do they want the generalized school readiness
Mm-hmm.
program, which we talked kind of went over a lot? So
Mm-hmm.
to control the education of the U.S. consumers, that's not true. I don't even have to look. I mean, he's talks about dumbing it down,
So
unfriendly power, but it's not about like government control. I don't think.
so what you're looking at, those are just the questions, but let's look for where he actually says, like the
Oh, okay.
answers, right? So I think it will be like maybe in the last paragraph. Like it'll have to be after the questions, right? Oh, I think I see it.
Was it here after this? I see generalized school readiness again.
Yeah. But look for so they're saying promoters, right? Look for that.
Oh,
Promote.
promoters wishing to keep their business financially solvent.
Oh, okay. That's you know, for that could be one of the things. But let's look for maybe like somewhere else because those are saying like possibilities. But let's look for like actual like what? Yeah.
Okay, that talks about just needs in here.
Uh huh.
Uh,
Did you?
oh, sorry. Can we go back?
No. You're in this on that sentence, and
Okay.
do you see that word?
They are
Promote.
directing it.
Do you see that?
They're directing it to the school administrators.
Yeah, because you see literature promoting, right?
Yeah.
Yep.
So I would say to control education of U.S. consumers, that's not why they're wanting to do it. I would just read the second one. I don't think that's right though. To defraud the schools of a great deal of money, it's definitely not that. Promote the success of a corporate sponsor. No, the answer is going to be D because that's what we just saw there.
Yeah. So you see how like it says promoters, you look for that word,
Yeah,
and
I
the
probably would
first
have.
one that came up did say the corporate thing, but then the second thing that came up was actually the one for like the school thing. Because remember the first time you saw promote like up there, it said, uh, yeah, are they promoters of? Yeah. Well, all right. I think you.
Yeah, I probably so.
Mm-hmm.
I probably, maybe I don't know what your approach is. Like, I'm willing to listen to anything at this point, but
Mm-hmm.
like, I probably would have read through these before I even referred back, just because I felt like I had a good understanding of it.
Mm-hmm.
Um, I knew right away that was a outlier. No, I knew that was wrong, so I would have gotten to here right away.
So I would have looked at. So they're saying the approach taken by promoters, right? In the question.
Yeah,
So I would have looked for the word promote. And the first time you saw it, like if you scroll up, remember the first time you saw promote, you see that. Like,
right here. Yeah,
and then, well, that's just questions. And then underneath,
yeah, because I didn't support anything.
yeah, then
Uh,
underneath, you see it says promoters wishing to keep their businesses financially solvent. That could lead people to choose C, right?
yeah, I could.
But it doesn't say that this is what the school readiness program promoters do. So that's why I asked you to look for promote again, and then you were able to find in the literature promoting their approach that is talking about the promoters of the generalized school readiness program.
So if I was doing this, I think I still would have chose D just because I feel like I had the context of the actual generalized school readiness program from what they had said,
Mm-hmm.
and I knew that I think I knew enough. I'm not trying to pull outside knowledge, but I knew enough from just the general sense of the passage that the
Yeah, yeah.
generalized school readiness program dealt with education and in no way helps corporate sponsorships because I knew that.
Mm-hmm. Yeah, I'm just trying to show
Yeah,
you, like, how, like,
but
that
like
other
if I saw,
path.
because that could happen.
Yeah, yeah.
I guess is it just more of a feel about whether I should have kept looking further in the like in the passage.
I would just look for promoter or promote,
Okay,
and
because
then
my first instinct was looking for that.
yeah, but they talk about that a lot, right?
Yeah, that's true.
So you wanna
They did say promoters
go
there.
exactly where,
Those just
right?
questions. So this was just questions. It didn't support.
Mm-hmm.
It didn't say what their wish was. It was just the author mainly, and
Mm-hmm.
then
And it's still talking about a general thing, right? It's not like saying generalized school readiness program.
but they didn't even talk about corporate sponsors though in the passage though. I mean, they did talk. That was getting into like corporate bureaucracy here, but like it didn't actually say.
Yeah, yeah,
So I feel like I would have still said that, even if
yeah,
I
yeah,
hadn't.
yeah. I
I
know.
think that was
Mm-hmm.
the answer was clearly though right down there.
Yep.
Okay.
So yeah, like I know you would have like gotten it right. It just that what you said about 90%, trying to make that higher.
Yeah, for sure. Because yeah.
Okay, let's see. The most reasonable inference from passage statements is that administrators are relatively reluctant to institute developmentally appropriate instruction because, all right, where do you think that would be?
So I would have probably read through the questions first, but
The choices?
so
Yeah, yeah. No, that's always good.
okay, inference involving the administrators. Well, the administrators was towards the middle, towards the end of the passage, somewhere in here. We talked about that was down in this area, I think. Unless I went too far.
So so it says developmentally appropriate instruction, so I would look for that.
Oh, okay. Well, he agreed that Piaget and them were doing that, so that was like
Well, that says
here.
intellectual development and stuff, but where does it
Okay.
say developmentally appropriate instruction?
Here.
It says that, and let's try to find if we can see instruction after that. Did you see it?
Not in this paragraph, unless I missed it. Is it in this paragraph?
Keep looking.
Okay.
Yep.
Okay.
All right. So now, what does it say about it? It's better, right? But it's hard to sell because of what?
Um, just because it's too difficult to implement, and it's not like it doesn't sell well to like the parents, and like
Show me like exactly.
um,
Because it says that the parents think that it's more reasonable.
so it's difficult to sell to the decision makers. I'm assuming the school board, like
Mm-hmm.
the
Mm-hmm.
the people, administrators,
Yeah, and why? Because they're concerned with what?
uniformity.
Okay. What does that mean?
I'm assuming that they're wanting some sort of a system that like basically can be applied universally to like the whole school system, but
Yep.
taking like a county by county approach.
Yes. Good.
Okay. Yeah, I would have tried to answer that before referring back.
Mm-hmm.
So, all right. So most reasonable inference is that the administrators were also listed why they were reluctant. Okay, it's they were by the parents. That's not true. It is. I mean, the parents did like the idea, but it doesn't answer the question. I don't think. Some untested theories. Oh, that's definitely not true. It is individualized, and therefore involves inconvenient process. So that's probably the answer. Promoted in slick presentations, and a C.
Mm-hmm.
Okay, yeah, that one wasn't too bad. That was pretty easy question.
All right, but you see how we're like look for the exact like word that they use in the question stem, and then you'll know where in the passage.
Yeah.
Okay.
All right.
Oh, this is an analogy. This is like old SAT stuff, like old SAT.
I never even took the essays. Yeah, I just went straight
Oh,
to community college.
ACT. Oh, interesting.
I studied for it, and I just kind of like was annoyed by it, so I just like whatever.
Yeah.
One can infer from passage that teaching in the generalized school readiness program, which is probably why I don't have a lot of standardized test experience, because I just could not focus instead for that. I did not care.
Yeah.
Because I didn't go straight. I'm kind of a non-trat. I didn't go to school until I was like
Mm-hmm.
23, I think. Like I
Yeah.
just kind of like work. Yeah. Generalized school readiness program is to develop is to. Okay, I know what they're doing. So they're comparing the teaching that he wants to happen in schools versus the generalized school readiness program as it is. So breaking farmland with a hand plow. This is a situation where I wouldn't really need to refer back to the passage if I had a good grasp of what these two things were.
Well,
Right.
which is the thing that is bad?
The generalized school readiness program.
Uh-huh. And that and which and is it like uniform or individualized?
It's uniform.
All right. And then the developmentally thing, that's the one that's good and individualized, right?
Yeah.
Mm-hmm.
So bad to good basically. So breaking farmland with a hand plow is to breaking fields by riding on a mechanicized cultivator. I think that's the opposite, because if they were saying A to B, the generalized school readiness program,
I think you're right. Yeah.
yeah, so I would say that's not true.
Mm-hmm.
Sending a yearly form letter to one's acquaintances is to sending personal notes. Like, well, the personal approach is approach one, and so it's the opposite.
Are you
I
sure?
goes past. So the generalized school readiness program is the one the less. This is the more general like approach. So that's why I eliminated that one because it's the opposite. The personal approach is developmental.
Mm-hmm.
So I would eliminate that one.
Wait. Why though?
Because
Isn't it?
so the general. I mean, I didn't finish reading all of it. Like sending your, if we're saying in like subsequent, if we're saying like in order, generalized school programs A and developmental appropriate is B.
Mm-hmm.
Sending a yearly form letter to one's acquaintances is to sending personal notes to one's friends. I just thought that has it backwards, like the order.
Wait.
Because we're saying the generalized school readiness program. Oh, so they're saying acquaintances though.
And a form letter, right?
Oh, I read that backwards.
Yeah.
No, I see. So general. Sorry, I read that backwards.
Mm-hmm.
Okay. Casting a line into like the fishing rod, fishing boat. That's no.
It's kind of backwards too, right?
So the family meal at home though would be kind of more personal though, like the developmental program.
Well, it's just cooking one meal, right? As opposed to a variety of dishes for each individual customer.
Okay, so
It's kind of backwards too, right?
yeah, so variety. You don't have variety with the generalized school program. Okay. Yeah, I might have had to go back to that.
Mm-hmm.
Like I definitely would have gotten rid of that, and then I would have been stuck on these two.
Yeah.
I had to read a little bit more, but yeah, that
But
one
you
makes
see
sense.
how it like connects to what we did before with uniformity and individualized?
Yeah, sometimes it's hard to keep my thoughts in order.
Yeah. So maybe we should have or it I don't know if there's anything like to highlight there, or if you like would have like written something down, like like just something like generalized school readiness program, like uniformity and developmentally appropriate teaching, individual.
Yeah, I probably was just reading a little bit fast because I had it in my head and I kind of like lost it because I was thinking personal. I was thinking that this one wasn't personal. I was confusing the order.
Mm-hmm.
Okay.
That makes sense though, right? How we did that?
Yeah, no, I understand that.
Got it.
That's another thing sometimes I read a little too fast sometimes because I'm worried about time.
Mm-hmm.
And I know that can throw me off. All right. Performance of high school students on examination. Noted for reliability and validity. That was up here.
All right. So now, yeah, this is like where we do the thing where it's like we know that when we first read it, it's like, okay, we get it. There's an example. We'll come back to it when they ask us to look at it, like reasoning within the passage kind of thing. These are like the grunt work, questions.
This is just where he was saying the researchers should make their own tests if they don't have one already in place.
Mm-hmm. But they want one that's reliable and valid, right? Good. Yeah.
Okay, so others' views on such tests suggest that scores could also be used to evaluate what. So this is like an outside the context one. Like where you try to apply it to like a real world situation. So.
Oh, like reasoning beyond the text, yeah.
Yeah, that's what I meant to say. Yeah.
Mm-hmm.
Usefulness of high school curriculum. Honesty of the gradings. I'm gonna go back. I would go back some a little lost with her.
Mm-hmm.
Appropriate instrument. Trying to make sure what they're saying the instrument needs to be. I mean, there's a reliability. I mean, I know what reliability and validity is. If the research believes the existing test is adequate. I don't know about the author's views on such tests, suggest that scores could also be used to evaluate the usefulness of high school. I'm not sure about that, so I would move on. The honesty of the grading in high schools, that doesn't have anything to do with the test. Appropriateness of the testing principles. Probable income of the students in later life. I don't know why. I'm going to go back to read the question. The performance of high school students. So they're saying we know that their performance in high school is reliable and valid. So what can we use that? So I need to read the question. Yes. So it's used to predict their success in college. What else could we do with that info? Okay. We know that. I don't know why it would use the usefulness of high school curriculum. We're talking about how they perform in high school impacts how they perform.
Are you sure
in
though?
College.
Is that like read that closer? Is it talking about how they perform in high school?
The performance in high school is used to predict their success.
Is it performance in high school or
is their performance after in college? Wait. No, it says that it's noted for reliability and validity.
So what though?
Their scores in high school.
Wait. So all right. Try. So that's where. So try. Yeah. Think about that. Is it saying their performance in high school or?
What do you mean? Like.
So it says the performance of high school students on an examination, on an exam.
Well, I mean, I feel like what they're saying is their performance on the exam reflects how they're going to do on exams in college.
Exactly. Yep.
I just don't understand using these. It's also used to evaluate. So, like, they're basically saying if you perform well on a test at point A, which was high school, you would do well in your next education as far as exams go. Mm-hmm. So it has to be like two things that are similar enough. So, I mean, I guess that might be helpful with the usefulness of a high school curriculum because.
This is kind of meta because it's kind of like talking about, you know what I'm talking about? Like the MCAT.
Maybe that would be,
and then
maybe this would be useful. Maybe that would be useful. I'm just at maybe right now. That might be useful because but I don't know. That just has to do with the way they teach it. It's just saying how well they do on their test.
Yeah. So it's almost like.
Well, I guess like.
It's almost like the MCAT and then the classes, like you were saying, right?
I don't know what appropriateness of the testing principles has to do about the passage, though, because
I mean,
with the questions, I don't think that one's true.
Mm-hmm.
And like seeing income, I don't. They're trying. I don't know. They might be trying to trick you because they're saying success to income, but I don't think you can make that connection because income and academic performance are two separate things. So I would probably eliminate that and go back to A and just like reassure I was right.
Yeah.
I guess if they found that you were good, like if how you do on a test in school impacts how you do on curriculum or your college tests, your success to that. I mean, there's probably a way they could use that information to figure out if the school curriculum's working. So A would be the true answer.
Yeah, because if you're able to, if a test can predict how you're going to do in college, then like it doesn't matter. Like then like if the test is used to predict how you do in college and not what you learn in high school, then it brings into question how useful is what you learn in high school. If the test is it's kind of like the MCAT, like how useful is it to like have like whatever, like biochem PhD or whatever in this, or like in this situation it's like SAT and stuff like that. But I'm just using real life ones examples or whatever, but yeah, basically.
Yeah. Okay. Yeah, that was a little tough for me. I think once I went back and read the first part, I think I was trying to focus on the stem of what the author was. I wasn't getting.
Mm-hmm.
I was trying to. Once I tied two and two together,
Mm-hmm.
it made more sense. For sure. Oh, I guess we finished the passage. All right.
Mm-hmm.
Cool. Yeah, that was helpful going through cars. Like I feel like I could potentially get most of them right.
Yeah, if you like start off like that way, where you're just really, really, really getting to the heart of what's being said and knowing like exactly what the words, like the nuances of the words are, and you know exactly why, because most of the time you chose the right answer, but now you know why every single other answer is wrong, so that boosts your certainty in what the right answer is. And coupled with like we're gonna practice on like like when it says something in the question stem, looking for it in the passage and being able to find that, so you get to localize it and see it like immediately where to go. Like all these things, like you're starting like relatively starting out with some of these, and as you do them more and more and more, it becomes like internalized, and then you're just going to be able to do it faster and faster and faster. At some point it'll click, like the titration curve, and that inflection point, and then you'll be able to do it like instantly.
Yeah. All right. So yeah, I think cars might be for me just like maybe careless mistakes with like not referring back enough and finding the right place, and then just I think I'm pretty good about getting rid of distractors usually pretty quickly. I don't usually, that's never reviewing. It's always like I was between two choices.
Yeah,
I think that's like everywhere on this test.
That's why that's why I made it so we focused on like two.
So like if you were doing this at time, is that basically the approach you would use? Like
wait,
you just read through it, highlight a little. Like I felt like maybe I waste time with excessive highlighting, just trying to like.
Yeah, I see that in a lot of students, because there's just so much fluff there. Like first, like try to isolate the like the main idea, right? Which is like a reformer should do this, and then like because that stuff is like hypothetical, right? And then the author will eventually later in the passage bring about like an actual example, which in this case was generalized school whatever, and that type of stuff, all those details, that's only relevant as like they come up in the question stem. And when they come up in the question stem, like practice on like being able to find it in the passage. That way it's like, so like remember when it's like promoter or whatever, look for that word promote, because instead of like looking throughout the entire passage, you know exactly what to look for and localize it.
For sure.
So like,
I think sometimes
over time you'll internalize it and you'll just get faster and faster and faster, so it'll be automatic.
Yeah, sometimes too, depending on the passage, like certain types of passages are harder for me to comprehend. I'll like get through. I'm like, oh crap, what did I just read?
Yeah. So like next time we'll do like those types of passages, like the ones that are like art history and like philosophy. Those types of passages are very, very. That's why like I focused on like nuances and words today, because in those types of passages there's extreme amounts of nuance and subtleties in the words that are used.
All right. Outside of,
Mm-hmm.
I'm running out of car stuff to do. Like I did all of the UWorld cars. I've done I've done 2/3 of the AMC cars, so I'm trying to think like what I should do now. I
Um,
mean,
you could do like start like you know like start doing them like very slowly like we did it, and like really 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 understand why something is right and why something is wrong. So you said that you have like a third of the car stuff left.
this is all I have left aside from I
Oh
think
wait,
there's
you said it's 13 out of 120 answered.
yeah.
Okay, so you have like over 100.
Yeah. That's all I have left. Is I think there's some cars questions in the guide. There might be like 20 in there.
Okay, so
Um,
why don't you do like that's still plenty. So you could do like two passages before we meet like next, and like really like do them like slowly. Like as your test approaches, then we're gonna like kick it up. So like
okay.
like
I don't think there's anything like major like I have to change aside from maybe paying more attention to certain details and the way I like go back to them. Because like I don't think just like cramming out like not cramming out, knocking out tons of cars hasn't made my score go up that
yeah,
much.
that's what
So
I'm
yeah.
saying. So like practice like those skills. Like look for the word. Like try that. Like you can see on my YouTube, I just had a student right before you, and we went over cars, and I would like test her like by saying like I would like look for a word, and I'd say, show me where that word is. Show me where this word is. So we're gonna do that also like next time.
Okay, cool.
Cause like
Um,
yeah, you'll get to a point because like I am at the point where I know exactly where the word is. I can like find it very quickly. So we're gonna work on that. So you'll get to the point where you can just like instantly see it. Boom, boom, boom.
should I try like not highlighting maybe, or just like maybe highlight something that stands out to me, but just try to keep it
Yeah, do like
minimum?
very minimal highlighting. Like I generally highlight to separate the like arguments, the points, the main points from the examples. You don't
Okay.
want like to get you know um, bogged down by the details of examples that they use.
And one thing I was wondering too about cars is like I did initially go very slowly, which does significantly improve my accuracy. But once the time kicks up, is where I start to be more careless and just have to like blast through.
Yeah.
Um,
So like
so like maybe I could. I don't know if I think generally I read a passage in about 4.5 minutes or so.
yeah.
I don't know if I'm reading too quickly or if I'm just I do know I stall on answer choices for too long. Like I'll
Yeah.
knock out two and then I'll be there for like 2 minutes, like crap.
Yeah,
Some of them
yeah.
I'll knock out in like 30, 20 seconds or something.
Yeah. So we're going to like specialize on that in that like
Okay.
for the since we have like what is this like 107 questions for cars, right? And
Yeah.
like a passage has like roughly like what like 5. So you'll
Yeah,
have
5 or 6.
so before I see you next, you'll have basically like 97 more questions. So we're going to like do like a progressive overload kind of thing. I'm just using
Gotcha.
exercise now. You see? So
Yeah.
so and things are going to change. Like it's not going to be like you're going to get faster at finding the words. You're going to get faster at separating like the main idea, arguments from the examples. You're going to be like you're going to just get faster, but it starts with like correct form, right? like
Yep.
like if you're like deadlifting or something and your form is crap, as you start to lift heavier and heavier weights, that like incorrect form is going to, you're gonna like snap like
Yep.
your vertebra or whatever. So it's like starting off very 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 like with correct form, very like it can be kind of like I know like a lot of my students like feel at first like, like, you know, we're taking too long, or like we're not doing enough like questions, and like on the real test it's gonna be like, you know, time crunch and stuff like that. But if you can get this foundational stuff like really, really down, then, and we're going to ramp it up each time, like again, like I'm gonna like be like, show me where this word is, show me where that word is. You're gonna practice. Your eyes are going to like adapt. You're gonna be able to quickly see things. Like you're gonna reach a point where you can like read with your eyes. Because a lot of people, most people, they read by reading out loud inside of their head.
Yep, so I do it.
Yeah, that's called subvocalization, and like the fastest that can happen is like the fastest that you can read out loud, which is like around like 250 words per minute. But if you can get to the point where you can like read with your eyes, or at least scan with your eyes, right? You know, it goes up like, you know, like 1,000-fold, because you're just going to, and you won't have like distractions either. Like if there's like some type of noise or something like that, since you're using a different part of your brain, like you're reading with your eyes instead of like using like your auditory like system. So that's going to speed things up as well.
Okay, cool.
All right.
Where we at time wise?
This is 2.5.
Okay.
So
are you done, or do you want me to do a little bit longer?
Let's see. One sec. All right, I think this is all I could do for.
Okay,
today.
You're good.
All right, so oh yeah, I didn't even get to go over the ones that you put in my notebook, but yeah, I'll like write some stuff down there. Okay.
All right, sounds good.
All right. So
all right,
yeah.
2.5 hours. All right, cool.
Yeah, and I guess I'll send you an email with like a reiteration of like the things I was saying now, like to practice on for like the next 2 CARS passages that you do.
Yeah.
Okay.
Yeah, this was good. We went pretty in depth. That was good.
Yep.
All right. Thanks.
All right. No problem. Have a good one. Happy 4th.
You too.